Okay, good evening, students. In today's lecture, we are going to talk about three phenomenons which are very important in the genetics. That is incomplete dominance, codominance, and overdominance. My name is Mrs. Mukta Kothari, and I am working as an assistant professor at Modern College of Arts, Science, and Commerce, Shivajinagar, Pune. So let us begin with our lecture. So, the first phenomenon, that is the incomplete dominance. Here, we all know that what is dominant allele and what is recessive allele. So, dominant allele is the allele which can express itself even in the presence of recessive allele. And recessive allele, it will be expressed only when exactly it will express? It will be expressed only when it is in a homozygous form. Otherwise, it will be masked in the heterozygous form. Its effects will be masked by the dominant allele in the normal condition. But in the incomplete dominance, what happens? The dominant allele, it is not able to mask completely the effects of recessive allele. So here, the dominant allele will completely mask the effects of the recessive allele, and as a result, what happens? Whatever progeny you will get, it will show the intermediate of the two parents. So as you can see over here, I have given an example of the snapdragon flower color. So here, if you cross the white color flower species with the red color flower species of snapdragon, the F1 progeny, instead of showing the red flowers, in normal condition it should show red flowers because red is dominant over white. But instead of that, what happens? It shows pink color flowers. Okay, so why this happens? Because it shows the incomplete dominance. So here, the red color in the presence of recessive allele in the heterozygous form, it cannot fully mask the activity of the recessive allele. And that is why as a result, the F1 generation will show the pink color flowers. Moving on, if you will see the F2 generation. So what can be the result for the F2 generation? If we will do the self fertilization of the F1, that is, if you cross the pink colored flower species with the pink colored flower species, then as a result, you will get the ratio of 1:2:1. That is red, pink, and white. Okay, if you will see, so 1:2:1 ratio you will get. Both the phenotypic and genotypic ratio I'm talking about. Okay, so everyone is with me. You are clear with this concept, I guess. Because in normal condition, if there is no incomplete dominance, so what happens in the F2 generation? What you will get? You will get 3:1 ratio, phenotypic ratio. Okay, but here in the incomplete dominance, you will get 1:2:1 ratio. So the phenotypic and genotypic ratios are same in case of the incomplete dominance. Now, moving on to the next phenomenon, that is the codominance, the second phenomenon. So here it is again different from the incomplete dominance. Here, what happens? The dominant and the recessive alleles, okay, in the heterozygous form, they are expressed together. Okay, so they will not show an intermediate, but both of them they will be expressed together. So here I have given an example of the rhododendron flowers. So here, if the white color flower species, it is crossed with the red color flower species. So the F1, in the F1 you will get the flower color which is combination of both. So both the red and white are expressed together. Okay, so here you have to remember that it is different from the incomplete dominance in a way where it will not show the intermediate of both the parents, but it will show the expression of both dominant and recessive allele together. Okay, so here in this case, both the white and red color are expressed together. I guess I'm clear with the second concept also. Let's move on to the third concept, that is the overdominance. Now, overdominance, it is totally different from the incomplete and the codominance phenomenon. Okay, here what happens? Uh, the heterozygous form. Okay, so the heterozygous form, it will have an advantage over both the homozygous forms. Okay, so here it will show the heterosis phenomenon. Okay, I have given an example of sickle cell anemia heterosis condition. So we all know that when the sickle cell anemia patient, what happens? What exactly is wrong with them? Their RBCs are sickle shaped. Okay, and why their RBCs are sickle shaped? Because there is a mutation in the beta chain of the hemoglobin, and because of that, their RBC appears sickle shaped. Okay, and the oxygen carrying capacity of those sickle shaped RBCs is very less as compared to the normal RBCs. Now, here, if you cross the homozygous, that is, if a person whose both alleles are normal, okay, if that kind of person is crossed with the person whose both alleles are showing the sickle cell mutation, in that case, the F1 progeny will be the heterozygote, okay, so which will show one normal allele and one sickle cell anemia mutation allele. Okay, so in this condition, what happens? This heterozygote, it will show the less deleterious effects. The physiological effects of the sickle cell anemia will be minimum in the heterozygote form. And secondly, second advantage is it will show resistance towards malaria. Okay, so but if you look at the parents, okay, so homozygous normal parent, it cannot show the resistance towards the malaria. And the second parent, which is recessive for both the sickle, or which is showing the mutation for sickle cell anemia in both the alleles, then in that case it will show the deleterious effects of the sickle cell anemia, very severe condition of the sickle cell anemia. Okay, so in that case the situation is really difficult for that person. It needs transfusions, blood transfusions. Okay, so but the heterozygote over here, it will have both the advantages. Okay, so it will have advantage, that is, it will be partially resistant towards the malaria, and it will also show the less deleterious effects of the sickle cell anemia. Okay, now why it will show the partial resistance towards the malaria? Because we all know that the malarial parasite, okay, so it feeds on the hemoglobin which is present inside the RBCs. So they will enter into the RBCs at a particular stage, and then they will start multiplying themselves into the RBCs. Okay, but if the RBCs are sickle shaped, they cannot enter into the RBCs, and hence the heterozygote will show the partial resistance towards the malaria. Okay, so I hope I'm clear with all the three concepts. Thank you so much for the patient listening. And if you have any doubts, you can always contact me on the given mail ID, that is kotharibiotech at moderncollegepune.edu.in. Thank you so much for your patient listening. If you have any doubts, you can ask me right now also. You have any doubts regarding these topics?
No, ma'am.
Okay. Thank you so much for patient listening. Thank you.